The overall goal of this procedure is to determine the mutation frequency in hematopoietic stem and progenitor cells and their differentiated progeny. First, isolate genomic DNA from FACS-purified bone marrow-derived cells of transgenic mice that harbor a LacI gene reporter of mutation events. Then package the individual LacI transgenes in a phage and infect E. coli. Plate the phage-infected E. coli on agar. Culture overnight to produce either blue plaques, indicating mutant LacI transgene, or translucent plaques for wild-type transgene. Then determine the number of blue versus colorless plaques. Replate the blue plaques to confirm that they represent true mutants and calculate the mutant frequency. Ultimately, sequencing the LacI transgene obtained from the confirmed blue plaques is used to identify the nature of the mutation to obtain a mutation spectrum for each population of interest.
So although this method is being demonstrated for hematopoietic cells, it can also be used for other tissues. The only thing that you then have to take care of is to see whether the temperatures for DNA digestion need to be adjusted. Demonstrating the procedure is Siming Chen, a senior research associate from my lab.
Thaw 1 aliquot of purified cells and add 500 microliters of ice-cold DNA lysis buffer. Vortex for 3 to 5 seconds on medium speed.